Hello everyone, welcome to SOS Classroom. So in previous lecture we discussed the cell structure and cell function. Different types of cell function we discussed, and now we discuss the cell cycle. How the cell cycle occurs. What is mitosis, what is meiosis, what is growth phase, mitotic phase, prophase, metaphase, anaphase, telophase. There are different types of stages and there are so confusions occurs during these stages. So we will discuss in very clear with the animations. So let's start. So first of all, discuss the what is cell cycle definition. What is the meaning of cell cycle? So when the new cell formation occurs, then cell has continued growth, then again function of the cell, then cell needs to die, then start again cell division. So this is called the cell cycle. Cell cycle means the starting from the immaturity to the mature and fully start the next cell divisions. So we'll discuss first of all what is the definition of cell cycle. Here you can see definition is the series of events. Series of events we discuss that growth 1, growth 2, synthesis phase, and cell division. Different types of cell division we will discuss. So, cell cycle is a series of events that take place in and leading to its growth, DNA replication, and division into two daughter cells in a mitosis stage, mitosis division, and in a meiosis stage, there is four daughter cells. So, this is graphical presentation of the complete cell cycle. So cell cycle occurs in the two phases. Number one is the interphase and second is the mitotic phase or M phase. You can see this is interphase and this is M phase. M phase is also called a mitotic phase. Again interphase divided into three steps. Number one G1, gap 1 and growth 1. S means synthesis or DNA replications, and G2 means preparation for the mitosis. So there are three phases completed in the interphase. After the interphase, there is a starting of the mitosis and meiosis. So mitosis occurs in the all over the body and meiosis occurs in the gametes, sperm in the males and ovum in the females. We will discuss in a detail. So this is the cell cycle. The cell cycle is divided into two stages, interphase and mitotic phase. In the interphase, there are three steps or three stages. Growth 1 means G1, gap 1. S, DNA replication or synthesis phase. G2 means again gap 2 or preparation for the mitosis. Then start the mitosis in the all over the body cells and meiosis for the gametes or germ cells in the male and females. So what is cell division? Cell division means the process by which a single cell divides into two daughter cells. Two daughter cells divide in the mitosis and four daughter cells in the meiosis. Mitosis means there are two identical daughter cell division, but in the meiosis there are four unidentical cell division. Unidentical means there are no comparison with the parent cells. There are no copy of the parent cells. But in the mitosis, the daughter cells just a copy with the parent cells. So cell division occurs in the two types, there is mitosis and meiosis. Mitosis also called a equational division and meiosis called a reductional division. Equitational division means the mitosis occurs from a one cell and divided into two identical daughter cells, just a copy. That's why it's called a equitational division. But in meiosis, there is reductional division. Reductional division means diploid cells change into haploid. Diploid means the cells carry 46 chromosomes divided into 23 chromosomes. That's why it's called a reductional. So equational division means division of the somatic or body cells. All over the body cells divide by the mitosis, and germ cells in the male and female divide by the meiosis. Mitosis called a equational division and meiosis called a reductional division. In the mitosis, one cell with the carry of diploid change into two identical diploid cells. But in the meiosis, one diploid cells change into four unidentical haploid cells. So we will discuss in detail. So before actual cell division, a cell undergoes cell cycle consisting of growth phase and also called interphase, mitotic phase or Anaphase. So these are the another names you can understand. Growth phase means interphase. So interphase means preparation phase. Preparation means growth of the cell and DNA replication occurs in the interphase. In the mitosis, mitotic or Anaphase, actual cell division. Actual cell division means mitosis and meiosis. We already discussed regarding this graph. Interphase divided into three stages, G1, S and G2. G1 means gap 1, S means synthesis phase, G2 means gap 2. G1 also called a cell growth, S also called a synthesis means DNA replications, and G2 gap 2 also called a preparation for mitosis. In the mitotic phase, divided into four stages, prophase, metaphase, anaphase, telophase. So these are also called a karyokinesis. Karyokinesis means division of the nucleus. And last is the cytokinesis. Cytokinesis means division of the cytoplasm. Cytoplasm division means the formation of the different types of organelles which are present in the cytoplasm, like mitochondria, Golgi apparatus, rough ER, smooth ER, lysosome. These are the organelles formation in these steps, cytokinesis. So we will discuss different types of graphical representations, and after graphical representation, we will discuss the mitosis. After that, we will discuss meiosis. So first of all, understand what is graphical representation to understand easily the mitosis and meiosis. You can see cell cycle. Cell cycle divided into two phases. Growth phase, also called a interphase. Mitotic phase, also called a Anaphase. So first of all, you remember the cell cycle divided into two main phases. First is the growth phase, another name is interphase. Second is the mitotic phase, also called a Anaphase. Growth phase again divided into three phases. G1 phase means gap 1, also called a growth. S phase means synthesis phase, also called a DNA replications. G2 phase, also called gap 2 or preparation for the mitosis. Next, mitotic phase, also called Anaphase, divided into two, mitosis and meiosis. Mitosis has two criteria, karyokinesis and cytokinesis. Karyokinesis has four stages: prophase, metaphase, anaphase, and telophase. So these are the division of the nucleus occurs. That's why it's called a karyokinesis. And last is the cytokinesis. Cytokinesis means division of the cytoplasm, means formation of the different types of organelles which are present in the cytoplasm. Next is the meiosis. So meiosis again divided into two phases. First meiosis first and second meiosis second. So first and second meiosis occurs. Some cells of the body cannot go from growth 1 to synthesis phase and directly change into the G0 phase. G0 phase means there is no further division occurs these types of cells inside the body. For example, some eye lenses cell and neurons or nerve cells inside the body cannot grow or divide inside your body. This is why eye lenses and nerve cells have G0 phase. means it cannot divide from one cells to another cells. So these are the graphical presentations. First of all, cell cycle divided into two phases, growth phase and mitotic phase. Growth phase divided into three, G1, S and G2. Mitotic phase divided into two, mitosis and meiosis. Mitosis done for the all over the body cells, but meiosis only for the germ cells in the male gametes and female gametes. Male gametes is sperm cells and female gametes ovum. Mitosis again divided into two categories. First is the karyokinesis and second is the cytokinesis. In the karyokinesis has four stages: prophase, metaphase, anaphase and telophase. And the cytokinesis, formation of the cytoplasm, means all are the organelles which are present in the cytoplasm form in this stage. I hope you understand this cell cycle. Next, first of all, we discuss the cell phase, means growth phase, interphase. After that, we will discuss remaining. In the growth phase, we will discuss the interphase, growth 1, synthesis phase, and growth 2, preparation for the mitosis. In the G1 phase or gap 1 phase, what will happen? During this stage, the cell grow in size. Cell grow in size means the newly formed cells enlarge in the size. You can see this is newly formed cells, then change into large size, then again change into the large size. So during this stage, the cell grow continue and cell increases in the size. Next increase is cytoplasm. Cytoplasm, because if the cell size increases, then cytoplasm also increases. Next, actively synthesize protein and enzymes, because different types of proteins and different types of enzymes formation occurs inside the cells. So this type synthesis of proteins and enzymes also occurs during the cell growth or increases in the cytoplasm. Next, preparation for DNA synthesis. DNA synthesis means preparation for the next phase. Next phase is the S phase, means DNA replications occurs. So preparation also occurs in this stage, but DNA synthesis occurs in the next stage. Some cells enter into G0 phase from the G1 phase and exiting all remaining phase. Exiting means there is no further phases occurs in the G0 phase. For example, cells of the islands and nerve cells. So these cells cannot divide and remain constant of the body. So islands and nerve cells. So this is the G1 phase. G1 phase means continued growth of the cells, continued growth of the cytoplasm, and continued growth of the different types of proteins, different types of enzymes, and preparation for the next stage. Next stage or phase is the S phase. S phase means synthesis phase. Synthesis phase means DNA replication phase. During this phase, what will happen? Centrioles and DNA replicates, and each chromosome duplicate into two sister chromatids. It means, first of all, DNA replications and centrioles formations, spindle fibers formations, and each chromosomes change into two sister chromatids. You can see these are the centrioles and these are the DNA synthesis. Next is the G2 phase. G2 phase means gap 2 phase. In this phase, cell continue grow. You can see cell continue grow and synthesis of different types of protein and enzymes, and check for the DNA errors. DNA errors means if the synthesis of DNA occurs, then G2 phase check the different types of errors occurs in the DNA. Next, preparation to enter into the mitosis. So these are the interphase, G1, S and G2. G1 means growth phase, growth of the cell size, growth of the cytoplasm, growth of the different types of protein and enzyme, and preparation for the synthesis. In the synthesis phase, synthesis of the centrioles, synthesis of the DNA replication, means formation of the DNA, and G2 phase continued growth cells, synthesis of the protein and enzyme needed for the cell division, and checking for the any different types of errors occurs in the DNA, and then prepare to enter into the mitosis. So these are the three phases occurs in the interphase. Now start the mitosis. In the mitotic phase, first of all we discuss the mitosis. In the mitosis, there are different types of phases, that is prophase, metaphase, anaphase, and telophase. Prophase, metaphase, anaphase, telophase is called a karyokinesis. Karyokinesis means division of the nucleus. And last is the cytokinesis. Cytokinesis occurs for the cytoplasm. Cytoplasm formation means different types of organelles formation occurs in this phase. So these are the two phases, karyokinesis and cytoplasm. Karyokinesis occurs in 4 phases and cytoplasm occurs in single phase. Prophase, metaphase, anaphase, telophase in karyokinesis and cytokinesis for the cytoplasm. So first of all, we discuss the prophase phase. What will happen in the prophase phase? Okay, you can see this animation. Then we will discuss the theoretical portion. You can see this is sister chromatins, this is centrioles, and these are the spindle fibers. So what will happen? Chromosome condense and become visible at pair chromatin. It means in the nucleus, the chromatin distributed as a thread-like structures, and when cell starts the cell division, then before cell division, the chromatid, which is thread-like structures, start the replications and form as a condense and formation of the chromosome. So first of all, chromosome condense in this stage and become a paired chromatid, which is also called a sister chromatid. Next, what will happen? Nuclear membrane start disintegrating. So you can see this is nuclear membrane and this is disintegrating. You can see this is integrating, means disappearing of the nuclear membrane or nuclear envelope. Next, centrioles move to opposite pole and form spindle fibers. You can see centrioles move to the opposite poles and formation of the spindle fibers. These are the spindle fibers. So this is called the prophase. So what will happen in the prophase? DNA condensation, formation of sister chromatids in the pairs, nuclear membrane disintegrating or disappear, and movement of the centrioles to the opposite poles and formation of the spindle fibers. So these are the steps formation in the prophase phase. I hope you understand what will happen in this stage. Next, start the metaphase. In metaphase stage, what will happen? The centrosomes align at the equatorial plates. Equatorial plates are also called a metaphase plates. And spindle fibers attached to the centromere of each chromosome. For example, you can see these are the chromosomes. These are the spindle fibers attached with the each chromosome. And alignment at the center, this is called metaphasic plate. You can see these are the alignment at the center, and this is called metaphase plate or equatorial plates. So spindle fibers attach with the chromosome with a special type of protein that is called kinetochore protein. So kinetochore protein helps to attachment with the spindle fibers with the chromosomes. So in this state, all are the chromosomes aligned at the center. You can see alignment at the center, and this is called equatorial plates or metaphasic plates. You can see animation. What will happen in the anaphase? Anaphase means separation of the sister chromatids. You can see separation and movement to the opposite directions. So first of all, centromere is split. You can see centromere is split and sister chromate is separate and move to the opposite pole. You can see these are the movement to the opposite pole and ensure each pole get an identical set of chromosomes. It means the 50% of the chromosome go into the right side and 50% chromosome go into the left side. Means equally division of the chromosomes. So this steps, you can see this is anaphase, separation of the sister chromatids and movement to the opposite pole. And next is the telophase. Telophase means formation of cleavage furrow. First of all, you can see this is called a cleavage furrow. You can see this is space between the both cells. This is called cleavage furrow, which will help for the contactile ring. And contactile ring helps to division of the cells or formation of the two cells. So chromatid change into chromosome and reach to the opposite pole. You can see reach to the opposite pole. Nuclear membrane reform and spindle fibers disappears. You can see another animation. Formation of the nucleus. Chromatid change into the chromosome. Nuclear membrane again formation and spindle fibers disappear. You can see these are the spindle fiber and now these are the disappear. And now formation of the two identical daughter cells. But there is no organelles present inside the cells and the organelles formation occurs in the cytokinesis. In the cytokinesis means cytoplasmic division. Cytoplasmic division means formation of the cytoplasm and different types of organelles formation in this occurs. You can see these are the different types of organelles formation in the cytokinesis. So these are the mainly 4 stages for the karyokinesis and 1 phase for the cytokinesis. So this is all about the mitosis cell divisions. In the mitosis, what we discuss, karyokinesis, there is division of the nucleus, and cytokinesis, division of the cytoplasm. Karyokinesis has 4 stages: prophase, metaphase, anaphase, and telophase. And in the cytoplasm, that is cytokinesis. Cytokinesis means formation of the cytoplasm, formation of the different types of organelles which are present in the cytoplasm. Next video, we will discuss the mitotic phase, meiosis cell division, how the diploid cells change into the haploids, and again haploid cells change into the 4 daughter cells, and which are the 4 daughter cells, haploid daughter cells. So we will discuss in the next lecture. So thank you so much and thank you for watching video. Click on the next video and we will see you there. And thank you so much.